Let me start off by saying welcome. My name is Jake Smith, and I am a grad student at Kennesaw State University. For the next 15 minutes, I will be discussing structure and diversity of bacterial communities in lead-contaminated soil. So the title of my poster is Bacterial Community Structure and Diversity in Different Levels of Lead-Contaminated Soils. So soil lead contamination is recognized as a global health problem, which can have adverse effects on human health. Because microbial community structure is an important index of soil quality, we ran a preliminary assessment on the microbiome of 3 locations in Georgia. These soil locations, Decatur, KSU Fuel Station, and Seartown have lead contamination levels of 20 parts per million, 90 parts per million, and 3,800 parts per million, respectively. The taxonomic classification levels of phylum and class was used because it best exemplifies subjective dissimilarity while limiting phylogenetic complexity. We hypothesized that as lead levels increase in the soil, number one, diversity of soil microbes would decline, and number two, diversity among the samples would increase. Regarding the methods, 2 subsamples were assayed from each location. Whole genomic DNA was extracted from 60 soil samples, 10 from each subsample. 5 samples from each of these extraction groups were pooled, a total of 12 pooled samples, 4 per sample location. The V3-V4 region of prokaryotic 16S ribosomal RNA gene was amplified and the fragments were sequenced. The OTUs were assigned and relative abundances were estimated. The data, which was analyzed with R version 3.6.2, was used to estimate the diversity of soil microbes within the samples. So the rare fraction curve in figure 1 shows that the curves of partial sequences of the 16S ribosomal RNA, where each curve represents, or should I say, estimates species richness. Because each of these curves are nearing the asymptote, it is safe to say that the sampling at each location is sufficient for estimating the microbial diversity. It is important to indicate the table at the top left corner shows 12 sites spread over 3 locations. Note that at the bottom right corner, there's another box that has the same sites, and each of these sites represents a curve. On the right half, you'll see figure 2 and figure 3. Figure 2 represents phyla, while figure 3 represents class. So both of these are analysis of variance of phyla and class levels, respectively, where the group means of the former are statistically different with respect to the Shannon Diversity Index. As you can tell with 2.32 e to the negative 5th p-value, the latter cannot be said when it comes to class because it has a p-value of 0.0594. Continuing, we'll look at figure 4 and 5. Both of these, figure 4 and 5, represent alpha diversity. They are stacked bar graphs of the phyla level, and the class level. The point to get across here is that compared to Cedartown, Decatur and KSU Field Station, seen right here and right here, show similar abundance profiles, especially with regards to cyanobacteria, acidobacteria, and proteobacteria. Below it is figure 6 and 7. Both of these are NMDS plots that have convex holes to help better visualize the beta diversity that's happening. The overlapping locations in figure 6 indicate possible relatedness between the phyla. As you can tell, there are 3 points of overlap, most of which is regarding the Decatur site. So D610F right there, D610B right here, and the very last one, which is the Seartown overlap, which is C15F. Figure 7 is a little bit different in that the class level lacks overlap. This is possibly because it is maybe indicating samples are relatively distinct. Um, Figure 8 is a quite a complex figure. So this is a dendrogram heat map. So it takes a heat map and a dendrogram and combines it. So if you see on the very left, you'll see the actual 12 sites. Above, you'll see and to the other side, you'll see the dendrogram part. In the very middle, you'll see the heat map. On the very bottom of each of A and B, you'll see a list of all of the respective taxon. For A, it's about um, phyla, and for B, it's about class. So to get that out of the way, let me just go back and start where the lighter colors, as you see right up here on the very left, on the very right of each one, they represent similar abundance values. So if we look back at figure A, this is generalizes the similarities and differences between certain groups, while in figure B, the class level, this provides more resolution of these similarities and differences, such as the distinction of similar group abundances within Cedartown, K2 Field Station, and Decatur. One thing to point out here, one major thing, is that there are two very large light areas in A and B. Both of these indicate similar group abundance across all three locations. And to summarize, the rarefaction curves indicate that sampling was sufficient for estimating microbial diversity. The Shannon index seen was greatest in Cedartown samples, which is opposite of our prediction. It was also significantly different at the phylum level, but not at the class level. Cedartown showed distinct distribution of some phylogenetic taxa, while Decatur and KSU Field Station had similar abundance profiles. Regarding the NMDS graphs or plots, the taxa in Decatur overlapped with the two other locations at the phylum level, while the class one didn't really have any overlap at all. The dendrogram heat maps, as we just saw, illustrate the similarities and differences among the locations. So, future directions. Regarding the Cedartown location, I would really like to measure the effects of lead contamination on its bacterial abundance and measure the presence or absence of the gene PBRA via qPCR. The reason I kind of want to specify on PBRA is that a lot of the studies that are looking at the diversity of lead contaminated soils, they usually look at PBRA because it is one of those genes that help the actual bacteria resist and thrive in toxic lead contaminated soils. In order to actually do all this, especially with looking at the specific gene I want to see, I need to compare these analyses with two other outgroups, so Decatur and KC Field Station. If the gene is present in similar numbers to the outgroups, I need to do a more intensive analysis. Good options that I have come across include like northern blot and sage. These two type analyses will help me identify how many of these genes are actually turned on. And finally, I want to talk about some of the acknowledgements. So this honestly wouldn't be possible without the help from Thomas McElroy, Sigurdur Greepson, and Marina Kother, who were awarded the NSF RUI grant for this project back in 2017. I would also like to thank Jaspreet Singh and Harry Vin for their part in the DNA extraction and PCR amplification. And I would also like to thank the LC Sciences for their work in helping us with the next generation sequencing. Thank you for listening to this presentation. I look forward to answering your questions on April 12th. Until then, have a great rest of your week.